On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Professor George Bassel from the University of Birmingham. And we're going to talk about a paper just published in PNAS, the title of which is Temperature Variability is Integrated by a Spatially Embedded Decision-Making Center to Break Dormancy in Arabidopsis Seeds. So thanks very much, George, for joining us again for a second appearance on the Arabidopsis Research Roundup. It would be great if you can give us an overview of this paper, please.
Yeah, great. Well, thanks for speaking to me today, GARNet. We were interested in understanding how it is that seeds integrate cues from the environment to time the termination of the dormancy. So we've known for quite some time that the hormones abscisic acid and gibberellic acid are involved in underlying a sort of an endogenous signal integration mechanism which seeds have evolved to integrate various signals, including light, temperature, nutrients from the environment. And that the balance between these hormones serves as a hormonal thresholding mechanism beyond which a seed, when, say, the dominant signal becomes the GA signal over the repressing signal of the ABA, the system will tip from the dormant to the germinating state. And in that way, we can have a thresholding mechanism using hormone abundance to control this developmental fate switching. So we've known this for quite some time now. This goes back to the work of Martin Corneef, who found these genes and really put molecular players to what had been inferred as endogenous growth substances back in the 50s by early plant physiology work. And we've known for quite some time that these hormones are involved in this process. We know much less about how these events are spatially embedded within the multicellular plant embryo. So what we did, actually my postdoc, Alec Topham, a very talented postdoc, went about in this BBSRC-funded project to create a large number of reporters which encode both the synthesis, perception, and response components to each ABA and GA. And using a quantitative 3D imaging platform to localize them at single-cell resolution within dormant seeds to understand how they're spatially distributed within a dormant seed. And resulting from this, what we found is a small group of cells located within the embryonic radicle, which turns out to be the mature root following germination. Mm-hmm. And that within this small group of cells in the radicle, disproportionate enrichment of all these components which underlie the decision-making process in seeds. What we did not expect, given the fact that there's an antagonism between each ABA and GA, which appears to occur at the level of abundance in the hormones. So as stated by the hormone balance theory, it's the relative abundance of each of these hormones that determines the developmental state of the system. So if you have more ABA than GA, it's a dormant seed, and when GA overtakes ABA in its abundance, it's a germinating seed. What came as a surprise to us was the fact that the cells which respond to each ABA and GA were not the same, meaning the ABA response and GA response did not spatially overlap in the same cell types. So it's in the vasculature that we see a lot of GA response, and in the root cap cells that we see a lot of ABA response, but they don't actually overlap in the same cell types. So in as much as the synthesis and degradation of each ABA and GA overlap with each of the respective response centers, the responses themselves are not cell autonomous. Okay,
that's cool. So, I mean, I urge people to look actually at the supplemental data of this paper. There is a beautiful figure which shows all these reporters, which, as I said, would have taken absolutely ages to put together, but of course is contained within one figure, but a lovely figure showing the expression of these reporters in the radicle using a Gus reporter line. So you've taken this information, you said, so spatially different data, and you've managed to put this together into a model to try and predict what's going on within the radicle. So can you tell us a little about the model and how that was developed?
Yeah, absolutely. So the idea is that it's through hormone thresholding in abundance that the system is switching fate from the dormant to germinating state. So together with my colleague Ian Johnston, a mathematician, the postdoc on the project, again, Alec Topham, developed a series of ordinary differential equations which modeled the interactions between the ABA response and GA response and their feedbacks back onto the gene expression would synthesize and degrade each of those hormones. So we created a model whereby we had each of the individual components. We had the receptor and the response, and then the feedbacks onto synthesis and degradation gene expression for each GA and ABA, and showed that this model was sufficient to create a bistable system. So in that sense, we created a model describing a hormone metabolic network, how the metabolic interactions between these hormones relate to one another. And we're able to recapitulate a bifurcating system which had each dormant and germinating states. So this was all very encouraging. So this was a very nice starting point of the project, but what caught us by surprise is the fact that not all of these interactions occur within the same cells, because we have non-cell autonomous response sites to each of the hormones. Okay. So in that, from that starting point of having that initial ODE model, what we went to do based on the microscopy data and the separation of the components was to embed these different interactions into a sort of a multicellular version of the model. So rather than having all of the components interacting with one another, we separated them into different compartments and then had those compartments communicating with one another, representing the movement of the hormones from the one site to the other. And from that, we found that, again, we got this bistability in the system, which was very encouraging. But what we found as well was the system, in fact, had some novel behaviors following on from the spatial embedding, which we didn't see when everything was present in the same cell. Okay. So this is where things got a little bit off-piste, in the sense that I was speaking to a colleague, a neuroscientist, and we were just talking about, I was telling him what we had found, and I thought it was very exciting. And he said, "Oh, well, that's just like the basal ganglia cortex loop, which is within the human brain in motor decision making. We have in our brains a spatial embedding of antagonistic signaling centers, not unlike what we're seeing in the radicle of the seed. So you have one signal which is inhibiting, which is the dominant signal. In our case, this is ABA, but a signal which is constantly inhibiting the impulse to initiate motor movement, and another center which is spatially separated from that, which is the positively acting one. And what happens here is there's a spatial embedding of these antagonistic signaling centers which are communicating to one another through neurotransmitters. Now, what's hypothesized in this context is that this spatial separation, because we're living in an environment with highly variable inputs through our sensory perception of the environment, that these inputs can be highly variable, that by introducing spatial separation between these components, that this can act as a noise filtering mechanism, meaning that you're not having everything hardwired and directly connected to one another, but by introducing a space, these fluctuating inputs, as they're communicating between the two centers, can be smoothed out just through the introduction of a delay in the communication between them. So this is what's thought to be happening in our decision to move and not to move. So we thought, well, I wonder if seeds, which also need to be able to time their decision to break dormancy accurately in order to ensure the survival of the resulting seedling, whether seeds are also using the spatial separation as a noise filtering mechanism as well. The idea is that if you do have rapidly fluctuating temperatures, that this would be smoothed out to increase the accuracy with which a seed can time the termination of dormancy. So what Alec did was give the seeds either continuous cold for 4 days, or give the seeds alternating cold and ambient temperature being 22 Celsius. And the idea would be that if the seeds were filtering the noisy inputs, that the inputs should be equivalent between the two, or in fact less in the oscillating version versus the continuous version, because the oscillating one would have less of an ability to break the dormancy because the oscillations are being filtered out, because it's a noisy input as opposed to a continuous one. To our surprise, it was in fact the oscillating temperatures that were more effective at breaking dormancy than the continuous temperatures. And this came as a surprise to us, and I mean, this is something that does exist in the literature. We've now demonstrated as well with Arabidopsis. The notion that a variable input is in fact instructive in guiding a developmental transition in seeds. So we have this idea of noise filtering, but in this case, the noise is in fact instructive and actually being harnessed by the seed. It's being used as a useful signal to time dormancy break.
It's absolutely fascinating that you've managed to break this down to this level of detail. And I think it's important to say that for people listening who might be using regular Arabidopsis seed that they've been growing at 20 degrees and just throwing onto plates and thinking, hang on, what do you mean about this variability and varying the cold and the heat? But you're working with seeds that will not germinate under regular conditions. They need a little extra to get them going, which is why you're able to tease apart the relationships in this system. So really, that's fantastic. That's obviously fantastic description of this paper, George. I've got to say, so you mentioned that just very briefly at the end. You mentioned about this analogy with the human brain, but it's very important to define that you're not saying that the plants have a brain. That is clear to say. It's just that the system is similar in the way that they remove noise in their system. Is that correct?
Absolutely. I mean, in no way do plants have brains. They would need neurons to have brains. Plants don't have neurons. They have other types of cells, a whole range of cell types, but none of which are neurons. So that is not what we're saying. But what I do see here, though, is a wonderful example of convergent evolution, where we have spatially embedded decision-making systems, which are similar in both the radicle of the seed and in the basal ganglia cortex loop in terms of motor decision-making. And I think it's a brilliant example of convergent evolution in these topological structures. Now, a key difference between the two is that in the human brain, this topological feature, this configuration, is being used to filter noise. But what we show using the modeling in our multicellular ODE model is that this topological, this separation, is in fact what is needed in order to harness the oscillating temperatures to break the dormancy. So it's through the spatial embedding that the variable inputs can lead to the phase transition from the dormant to the germinating state. So if we have them all in the same cell, we don't get that behavior. So whereas the human brain is filtering noise, the seeds, you can say, are amplifying or harnessing the noise by having the same configuration.
Great. Absolutely. Well, thank you very much, George, for joining us today. I think that's been incredibly informative description of the paper. Thanks very much.
Thank you, Ryan.